Hello, my name is Chen, and this is my presentation for 918's final project. It's on molecular gradients and their translation to transcription factors and their effects. Molecular gradients are something that's very important in development. Molecular gradients can include such things like transcription factors, hormones, and morphogens. The gradient encodes positional data that can be used to help determine cell fate or differentiation, regulation, as well as axonal and neuronal guidance, all of which helps develop the regionalization of our complex brain. An excellent example is to our right. When different transcription factor gradients of OTX2 and GBX2 interact with each other to determine the regionalization of the mesencephalon, or the midbrain, and the metencephalon, which is part of the hindbrain. We'll go into more detail on how molecular gradients accomplish this in later slides. The basic principle of how this is accomplished is fairly simple. The gradation of molecular gradients allow for positional data to be read. This positional data is then leveraged for differential regulation that gets translated into transcription factors. These transcription factors then go on to affect further downstream changes, be it altering gene expression, signaling another cell, or setting up another gradient. We'll examine the midbrain-hindbrain boundary and their differentiation, as well as the nasal and temporal axonal innervation of the tectum, to give you context on how this process works in the real world. The midbrain-hindbrain boundary, or the MHB, refers to the boundary between the mesencephalon and the metencephalon, respectively part of the midbrain and the hindbrain. It's an important boundary that sets up regionalization of later brain structures such as the cerebellum and the pons of the hindbrain, as well as the tectum of the midbrain, all of which have different functions. This boundary, highlighted in red, and the differentiation of the midbrain and the hindbrain is accomplished via, you guessed it, molecular gradients and transcriptional factors. In early brain development, the neurotube forms brain vesicles that can be characterized into the forebrain, the midbrain, and the hindbrain. These categories can be further organized into the telencephalon and diencephalon for the forebrain, the mesencephalon for the midbrain, and the metencephalon and myelencephalon for the hindbrain. These categorizations are characterized by distinct combinations of transcription factor expression. For example, the forebrain is characterized by the expression of PAX6 and OTX2. OTX2 is expressed in both the forebrain and the midbrain. The midbrain, though, expresses GBX2. GBX2 and OTX2 cross-inhibit each other. This leads to a distinct gradient with low concentrations of GBX2 and OTX2 between the midbrain and the hindbrain, the midbrain-hindbrain boundary, if you will. The special zone of low GBX2 and OTX2 induces the production of the morphogen FGF8. FGF8 will then go on to activate the RAS-ERK pathway in the surrounding cells. This leads to the differentiation of the midbrain and the hindbrain. Taking a closer look at the RAS-ERK pathway in this diagram here, you can see that depending on where the cells are in the gradient of GBX2 and OTX2, the activity of the RAS-ERK pathway is modified. Highlighted in navy, you can see that a position of high GBX2 activation of the RAS-ERK pathway leads to the production of the IRX2 transcription factor, which will lead to the making of the cerebellum, a hindbrain structure. Highlighted in orange, at locations of low GBX2 and OTX2, the midbrain-hindbrain boundary, the activation of the pathway leads to the production of the transcription factor P3, which facilitates differentiation into R0, or the isthmus. And finally, highlighted in cyan, you'll see that activation of the RAS-ERK pathway, where there's high OTX2 expression, will lead those neurons to differentiate into the midbrain by inducing the production of the transcription factor engrailed in PAX2. Hopefully, you can begin to see how molecular gradients and transcription factors play a major role in our development. These gradients and transcription factors not only play a role in differentiation, they also help in our axonal and neuronal guidance. An example of which can be seen in the development of the retinal topic maps of our visual system. Our visual circuitry is organized to have a topographic relationship. That means that positions of the neuron correspond with the positions of their terminal arbor in the target field. This organization preserves the visual topography in the brain and provides a neuroanatomical basis for contiguity. In other words, the position of our neuron is conserved in the position of its axonal innervation, and this allows for an anatomical encoding of positional information. This process of retinotopic maps is described by the diagram shown to the right. This retinotopic mapping can be best exemplified in Roger Sperry's experiment, where he rotated a frog's eye before retransplanting it. Because of the retinotopic maps, the retinal neurons reinnervated the tectum exactly the way they were used to. But since the eye has been rotated, the field of vision is inverted. What was once up is now interpreted as down. What was once left is now seen as right. In this video, you'll see how a normal frog reacts to a fly. It jumps towards it and has no trouble snapping it up. But for the frog that has undergone the eye rotation, you'll see upon presentation of the fly, the frog always moves the opposite direction to the fly. Now, this presents a question. How does the axon know how to wire the said retinotopic map? An experiment that helped deduce out how the axons were wiring this retinotopic map is the striped carpet assay from Walter and Bonhoeffer. They created an assay utilizing tectal membrane from both the anterior and posterior parts of the tectum that they organized in alternating fashion, creating the striped carpet. Then, with a retinal strip, they allowed for retinal neurons to innervate the tectal membrane. They found that the temporal neurons selectively innervated anterior tectal membrane, whereas the nasal retinal neurons wouldn't have any preference for which type of tectal membrane. Something must be influencing the temporal neuron's axonal preference. Either the anterior membrane is excreting an attractive cue, or the posterior membrane is excreting a repulsive one. Upon heating the tectal membrane, which would denature the proteins, they found that the temporal retinal neurons no longer exhibited preference. This suggested that the posterior tectal membrane was secreting a protein that was repulsive to the temporal axons. Bonhoeffer and colleagues, upon examining the choices that temporal axons made between membranes extracted from successively posterior tectums, discovered that the repulsive activity is graded across the tectum, highest at the posterior end of the tectum and lowest at the anterior. The molecules and corresponding receptor was discovered to be ephrin 1A as the ligand and EphA as the receptor. The expression of the receptor was also graded in terms of the position of the neurons in the retina. Nasal axons expressed less of the receptor and thus wasn't repulsed by the high ephrin 1A concentration found in the posterior end, whereas the temporal axons have high expressions of the EphA receptor and thus prefers the anterior end, which had a smaller concentration of the ligand. But how did the ephrin 1A gradient come to be? Remember back to the midbrain-hindbrain differentiation. The midbrain develops into the tectum. And what was the transcription factor that induces formation of the midbrain? Right, it was engrailed, otherwise known as EN1 and EN2. The transcription factor EN1 and EN2 induce the formation of the midbrain and thus the tectum. Thus, they're still about. In fact, transcription factor EN1 and EN2 can be found in the tectum in a concentration gradient, with higher concentrations of EN1 and EN2 in the posterior of the tectum and lower concentrations in the anterior. The same kind of gradient ephrin 1A is in. Coincidence? Well, what happened if you did the striped carpet assay with the temporal axons, but you would hit it engrailed with 4D9, a anti-engrailed antibody? You see that the temporal axonal lost preference, but if you rescue the engrailed with a 4D9 epitope, you'll see the preference return. And so we see that ephrin 1A's gradient, which regulated the interaction of the retina axons, is itself regulated by another gradient, that of the transcription factor engrailed. As you can see, different transcription factors, receptors, and molecules can play different roles in different pathways. Engrailed not only induced midbrain formation, but also regulated ephrin 1A. Although there's really two EPH receptors, EPHA and EPHB, there's many different ephrin ligands which can act in different ways. This schematic outlines how ephrin A and ephrin B can induce different effects. We have the ephrin ligand bind to the receptor EPHA or EPHB. This would then activate the Rho-A pathway. The Rho-A pathway under ephrin A induces the axon repulsion we see in temporal axons. Now, the Rho-A pathway under ephrin B, though, actually induces axon attraction. Ephrin B and EPHB and axonal attraction is actually the mechanism in which the dorsal and ventral mapping of the retinotopic map is wired. Hopefully, after this presentation, you have a deeper appreciation of how molecular gradients and transcription factors play very important roles in directing development. This is only two examples, and I hope you've gained a sense of how interconnected and influential molecular gradients and the subsequent transcription factors are in making sure our body develops according to plan. Thank you so very much, and I hope you have a good day.